Welcome to our presentation. We are here today to discuss a new description of the muscle architecture in the abductor mandibulae complex of the spiny dogfish Squalus acanthias. I am Natasha Nevu with my lab partner Renee St. Jacques, and we are from the Westfield State University Biology Department.
First and foremost, we'd like to start off this presentation by pointing out that the spiny dogfish is used in a lot of different comparative anatomy textbooks as a model organism for the fish species. What we'd like to do in this experiment is to kind of point out that there are a lot of different drawings and it's not very consistent of how the muscle structure is on the spiny dogfish. As you can see.
As Renee was saying, in each of these illustrations, which are just a few of the many that are out there, the adductor mandibulae muscle is just referenced as this big blob of muscle, and the muscle direction varies in many of these pictures, with the two pictures in the upper right not showing very much of the organism at all, but are being used as diagrams to represent their feeding mechanism. And it's very important to understand what is actually occurring here because this is an important mechanism. If they can't feed, they can't survive.
So for this experiment, the materials and methods we used basic anatomy dissection tools, manual manipulation to move the jaw in and out of the feeding position, digital and microscopic photography, and the muscle nomenclature we'll be using to describe the different parts of the anatomy in this presentation will be the Wilga and Moda diagram that you can see on the right that has all the different muscles labeled. So that's the nomenclature that we will be using.
And during our experiment, we also wanted to look at if there was a best way to look at the specimen, if a preservation method was the best over others. So we looked at fresh specimen, preserved specimen from a couple different companies. So we looked at bucket preserved specimen, and we also looked at vacuum sealed specimen.
So we will be focusing mostly on the QMS architecture, which is the protruding muscle on top of the QMA in the adductor mandibulae complex. We found that there is a tendon attached to it that goes all the way back to the top of the skull, attaching to the postorbitalis, which is something that is usually cut out.
Here's another view of this muscle architecture. As you can see, this tendon from an upward point of view attaches from the postorbital process down to the QMA. And just to double check that it is actually attached to the QMS, we cut the tendon close to the postorbital process and pulled it back. And as you can see in the picture on the right, the muscle fibers are going up towards this tendon, and there is a connection between the two.
So this is a first draft of what we would like to use as a diagram for spiny dogfish dissection. As you can see, we have completely sectioned out the abductor mandibulae complex into its different muscle parts, which is often not shown, as you can see from the previous diagrams we've presented. So we have the QMV showing, the QMA showing, the QMP showing, and the QMS showing, which we are proposing to change to be named the postorbitalis because it is attached to the postorbital process by that tendon.
And during our research, we looked at various diagrams, and we found one from Marion back in 1905 that shows that this QMS architecture is made up of various components going in various directions. They hint on this QMS muscle that attaches to the postorbital process and down to the QMA, but they show it both in a cut version and an intact version without putting an actual label on this, and it has not been seen in other textbook variations of this diagram since 1905. So that's over 100 years that we've been missing this accurate depiction.
As for the QMP, upon dissecting, as shown in the Marion figure, we have also found that the muscle fibers of the QMP don't lay flat. They're not horizontal like all the other muscles. They're actually vertical and they stick out to the surface of the skin. And we have hypothesized that it potentially either meshes with the QMA or there is a tendinous sheet over the top of it, which we might have found some evidence of.
So during the dissection process, I carefully skinned the shark and was able to keep this tendinous sheet intact. And we found that this muscle or this tendinous sheet connects to this QMP portion of the muscle that previously was thought to just end abruptly. And this makes more sense that it attaches to this QMP, but it also goes all the way up to the anterior portion of the cranium just below the rostrum, showing that there could be some sort of interaction between the QMP and a part of the rostrum that helps this organism in the feeding mechanism.
So I'd like to recall back to the different diagrams that we showed in the beginning of the presentation, and to really point out that if you look at the diagrams now after the evidence that we presented, you could see that they're not very descriptive at all. The abductor mandibulae complex is just a blob of muscle when it's missing all the different sections of the adductor mandibulae complex, which we would propose to add in to figures from now on. And we also propose to standardize a figure across all textbooks so that we wouldn't have this issue of inconsistency when teaching, which is obviously very important to have a consistent teaching method, especially in a model animal like this.
So, as we had mentioned before, we looked at various different preservation methods, and we found that the fresh specimen were some of the best options for dissection as their muscles remained intact better and longer. It was also easier to get the muscles to remain intact when we were doing the dissection, and they moved in a proper alignment that we would expect the tendons and muscles to move. As for the bucket preserved specimen, some suppliers were better than others. We found that the Carolina company was one of the better ones for bucket preserved specimen. They don't remain intact quite as long as the fresh specimen, but they were a very good alternative, where the Ward Science specimen were not as good, and the vacuum sealed specimen were some of the worst for observing the muscles because the skin is so tight on the organism, it is very easy to cut into the superficial muscles and tendons without noticing, and it just is not as great of a sample as, say, like the fresh specimen or the bucket specimen.
So upon looking at all of the data we collected and finding that there are multiple muscle sections in the abductor mandibulae complex, we would like to not only propose a new drawing of a standardized diagram, but we'd also like to bring up some questions and other thoughts, such as how does the new architecture of the abductor mandibulae affect the feeding mechanism, the kinetics and the mechanics of the feeding mechanism, the bite force of the animal, and things along that nature. Also, what is the best species for anatomical comparisons? Should we continue to use the dogfish, or now that we found out that its musculature is more complicated, should we choose another organism? Would there be another shark species that would be better? Would there be another fish species that would be better to show this because of how uncertain and inconsistent the dogfish diagrams have been? Would there be a better way to teach anatomical comparison classes with a better suited organism for teaching? And should we evaluate the methods of preservation for specimens used in classroom dissections? Should we move more towards buckets and less towards vacuum sealed organisms for dissection?
We would like to thank the Westfield State Biology Department for providing us this opportunity. A thank you to our fellow lab members, Matthew, Samantha, and Cassandra. And a final thank you to the Tri Beta Honor Society at Westfield State University for including us in their membership.